Okay, so welcome to all of you. Hope you guys had a good weekend. So we're starting a new unit, genetics, and it's going to be very different than our other units. Okay, before we had units like, or we went through biochemistry and then metabolism, which is very like on a micro scale, and it's be kind of courses that you would take in university called cellular biology or microbiology. Well, what we're going to go into now is genetics, which is very different than the other ones. And I'm going to kind of give you an introduction lecture. Now, there's no content on this part here, but it's very important for you to know because you're going to be tested on this in university. And I taught first year university, so I know exactly what you're going to be going into. So you need to know some of the background on how we even figured out in the first place that it was DNA that's hereditary material. So today's whole lesson goal is to go over 3 experiments that basically proved that it's DNA and not protein that is the hereditary material. Okay, the material that is passing on, genetic information. So we're going to go through a slideshow today. I'm going to have my slideshow posted underneath the bottom. There's a couple activities that I'd like you to do in there as well. You're going to log on to a website and go through some of these experiments a little bit more in depth than I go through. Okay, but the key thing is you're expected to know, how they did the experiments and what the experiments mean. Okay, in terms of proving that it is in fact DNA that controls our genetic information. So let's take this up. I'm going to make this full screen just so that it's easier to see some of the diagrams. And, you're going to have my slideshow with you as well. Okay, so here's our overview. Now, just to kind of a picture of, where DNA is. Here's our cell. So, any cell in your body, okay, with the exception of your sex cells, is going to have a complete copy of your DNA. Your sex cells are only going to have half of it. And then the other ones are red blood cells. They also don't have DNA in them. Okay, so we take a cell. And then inside the nucleus of the cell, it's not in the cytoplasm, but inside the nucleus we have chromosomes. Okay? Well, all chromosomes are, and you probably remember these from grade 11, is if you take DNA and wrap it around these little things, proteins, okay, and specifically they're called histones. If you wrap them around protein, that's what a chromosome is. It's basically just a piece of DNA that's wrapped around protein. Now we do this because DNA is a super, super long, like you think of how long this entire molecule is. Okay, we've got to fit this really, really long molecule inside not only our tiny cell, but the nucleus inside our cell, which is a very small area. So the way that we package it so that we can keep this really, really long, molecule or fiber inside our cells is we package it in chromosomes. Okay? So when it's packaged together and it's in chromosomes, we can't really do anything with it. It's kind of stored. And then when we want to do stuff with DNA, like make a protein or replicate it, we take them out of the chromosomes. Okay. So the important thing here is the discovery of DNA is no doubt one of the most important scientific discoveries to date. Okay? There is a huge future in genetics. Okay, we're going to talk about that in our last kind of chapter. We're going to go through some of the technology of genetics, but it's super, super important discovery. I mean, perhaps the most important discovery. Okay. Now, the next few slides, we're going to go over some of the history of how we developed our present understanding of DNA. It's important to know these experiments on how they actually discovered DNA. Okay. First things first, it's been known for thousands of years that organisms resemble their offspring. Okay? If you look at, so here's Demi Moore and here's her daughter, and you can see how much they resemble each other. Okay? They almost look identical. Or here's a father and here's a son, and how much they look the same. If you look at your own parents or you look at your brothers or sisters, you see similarities. Although you may want to hate to admit it, you're going to see similarities between the way that you look and the way that your parents or your siblings look. And it's not just looks, it's also behavior. Okay, genetics control a lot of our behavioral functions, so we also behave similarly between our parents and our offspring. So we've known for thousands of years. I mean, you can just see that parents resemble their offspring. Okay. But what was really eluding us was what exactly is transferring this information? What kind of molecule is being transferred from one form to the other? Well, if we go back to grade 11 and we went through Mendel was way before the discovery of DNA. Okay, he was just kind of discovering inheritance patterns and these laws that govern inheritance patterns. But what he did kind of figure out is the hereditary material is on chromosomes. Okay, well, chromosomes are composed of 2 things. They're composed of DNA, or nucleic acids, and protein. So therefore, is one of these 2 things that is going to be controlling our genetic predisposition for things. So we've got to figure out, well, is it protein that's carrying the genetic information, or is it DNA that's carrying the information? And that's what we're going to figure out today. We're going to go through experiments that proved it was DNA and not protein. Well, the first thing is, regardless of whether it's protein or nucleic acids, whatever the hereditary molecule is, it must meet the following things. Okay? It has to contain information to make enzymes and other proteins. So it's either proteins making proteins or it's DNA making proteins. But regardless, they have to be able to make proteins and enzymes. And the key thing is that when we're going to build a body or run a body, we need to constantly be doing reactions. Well, what's under the ultimate control of reactions is enzymes. Without enzymes, you have nothing. Right? If you want to make some skin, I'm going to have to do some reactions to make skin. So I'm going to have an enzyme that controls the reaction for making skin. So really, in the ultimate control of things is protein, and specifically it's enzymes. So we need to have whatever the genetic information is, it needs to be able to control and make enzymes, okay, or proteins. The other thing, it has to have the ability to duplicate itself without many errors. Okay, you're over 1 trillion cells right now, which means that you've had to replicate a single cell well over 1 trillion times. Okay, multiple trillions of times you've had to replicate it. And you have to make sure that when you replicate one cell, and say you make a new skin cell or you make a new tongue cell, you have to make sure that that cell contains an exact copy of your DNA error-free. Okay, or with limited errors. Now, errors do pop up, and that's where freckles are. Say your skin cell tries to make another skin cell, and there's an error in it, it might turn into a freckle or a mole. Okay, and hopefully that is localized within that area. If it's not, then you may have to get your mole looked at, because it could be, transferring that error into other cells, which is a form of skin cancer. And the last thing, we have to have the flexibility to permit a few random mutations to increase the variation in a population. So we have to have this genetic molecule, whatever it is, whether it's protein or nucleic acids, it has to be able to change slightly so that we can create variation. And by variation, I mean that we have to be able to change the protein or the nucleic acid slightly so that it can make people with blonde hair and people with blue eyes and people with red hair and people with green eyes, so that we can have some kind of mutation. And that's the importance of variation in a population. And we talked about that in grade 11, of how important variation is. We can't all be clones of each other, that it's horrible. Okay, so they originally said it's got to be protein. The hereditary material's got to be protein for the following reason. Okay, when we have proteins, we have 20 different monomers. And the monomers of proteins are called amino acids. So these are all the different amino acids. So this whole thing right here is a protein, and that's what a protein is. It's just a polypeptide of amino acids. We should know that from our first unit. We said, well, when I'm talking about protein, I have 20 different amino acids I can grab from, okay, in order to produce a protein. But if I want to create something using DNA, I only have 4 bases, A, T, G, and C. So because hereditary is so complex, it must be made by protein, because protein's got more options to be able to make more complex things. So they originally thought it's got to be protein. And they, for a long period of time, thought it was protein, was the hereditary material. It took a long time for them to originally figure out its DNA. And this is how they figured it out. It was DNA. Okay. An experiment performed by Frederick Griffith was instrumental in determining that DNA is the hereditary factor. Okay. Now this is key. You would want to highlight this if you have printed out my notes. The results of Griffith's experiments did not provide evidence for or against DNA being the hereditary material, but rather sparked an interest in another scientist, Oswald Avery. So Avery performed another experiment, okay? And he only performed this experiment because of the results of Griffith's experiments. So this is the idea that science is a building ground where Griffith did an experiment, Avery looked at his results, did a different experiment, and then our third one's going to be Hershey and Chase, did a third experiment. So the 3 experiments that we need to know are Frederick Griffith's, transformation, Oswald Avery's, and then Hershey and Chase. So we're going to start with Griffith's experiment, okay? So the first thing, Griffiths was from London, England, 1928. Okay, so we're going through the Great Depression right now. Okay, he was studying a deadly strain of bacteria killing thousands of people, pneumonia. Okay, so Streptococcus pneumonia, if you remember our naming from grade 11. Okay, the Streptococcus, so it's going to be a bunch of little circles in a group formation, and he's creating pneumonia. Okay, so what pneumonia is basically when a bacterial infection gets inside your chest cavity, starts replicating, and then fills up your chest cavity with mucus. So this is a person with pneumonia. They would do an x-ray, and you can see this is all filled. Their chest cavity is filling up with mucus. There's an empty chest cavity. There's one filling up with mucus. So this is the disease that he was working, or the bacteria that he was working on. So he was working on developing an antibiotic yet, but it wasn't invented yet. So at the time, people were dying of pneumonia. So if you've ever got pneumonia in 1928, you would have died from it. Okay? So he's doing several experiments with pneumonia, and one particular was interesting. And he called it the transformation experiment. So this is Griffith's transformation experiment. So he created 2 different strains of bacteria, an S strain, which was a pathogenic strain. And what that means is it could kill people. It was the bad strain. And then an R strain, which is very similar, but it didn't cause you to develop pneumonia. So we've got one strain of bacteria that causes you to develop pneumonia, one strain that doesn't, and they're very, very similar. Okay. So what this means is that the S strain contains genetic information that the R strain does not. The S strain contains genetic information on how to infect you and how to give you pneumonia. The R strain doesn't have that information, or it would give you pneumonia. So the next few slides are going to show what happened in the experiment. And I'm going to go over this kind of picture here. So this is our diagram, and we want to make sure that we understand this. Okay. So the first thing, he takes S cells, or S pneumonia cells. Okay, so these are the bacterial cells that cause pneumonia. He injects them into a mouse, and of course the mouse dies of pneumonia, because you just gave it pneumonia. Then he took R cells. Okay, so R cells are a different bacteria that doesn't cause pneumonia. He injected this bacteria inside a mouse, and the mouse is alive. Okay. So these two are just kind of controls. Third thing is he took the heat-killed S cell. So he takes this bacteria that normally kills a mouse, heats it up, and destroys the enzymes. So we should understand here now, we're going to destroy the enzymes of the bacteria. So now those enzymes don't work. And if the enzymes don't work, reactions don't work. So if the bacteria isn't functioning properly, it's not going to be able to infect a mouse. So that's why heat killing the bacteria. You heat killed them, you inject it in there, and the mouse is healthy, no problem. Okay. Now this last one was the most interesting. Okay. Is they took heat-killed S bacteria. So the enzymes have been destroyed. The protein has been denatured. But the nucleic acids, or the DNA, is completely fine, okay, inside these cells. These bacteria over here haven't been heat-killed at all, so they're completely functional. And what happened was when they injected both of these together, the mouse died. And when he died, they checked the mouse, and they found that there was living S cells. Before, you only injected dead S cells, but somehow the dead S cells was able to transfer genetic information to the living R cells and then transform the R cells into S cells. Okay, if that makes sense. You may want to rewind the video to go over that again. Okay, so that's why it's called a transforming experiment, because the S cells were able to take their genetic information, even though the cells in general were dead. They were able to take their genetic information and transfer it into another bacteria, transforming that bacteria that was R into S. Okay. Now there's a video down here that I would suggest watching. If you type in Griffith's transforming principle, you can get a bunch of different videos. Any of them are good. I found this one very helpful, so you could take that link and type it in if you want to take a look at it. Okay. So that's Griffith's experiment. So that's experiment 1. Okay. So the results of Griffith's experiment did not prove what hereditary material was, but it did show that one type of molecule is able to pass its information on to another bacteria, and that information was being passed on through a molecule. What molecule, we don't know. It's still protein or nucleic acids. So then Avery went on and did his experiment. Now I'm just going to briefly go over his experiment because I'm going to ask you to research it on its own. But he basically said, okay, I'm going to take the S strain, okay, so the virulent strain, and I'm going to take the S strain and take only the RNA from the S strain and see if we can get RNA to transfer the R strain to an S strain. And it couldn't. RNA wasn't able to do it because it can't carry genetic information. So then he said, okay, I'm going to take the protein out of S strain, take all the protein out, and put the protein of S strain with R strain, and nothing happened. If protein was the genetic information, it'd be able to transfer R strain. Then he said, okay, I'm going to take the DNA out of the S strain, put the DNA from the S strain with the R strain, and oh, look at that, it transformed it into an S strain. Just to confirm it, let's make sure lipids aren't transforming them. So I'm going to put lipids from the S strain in with an R strain, and there was no transformation. Okay? Then I'm going to take carbohydrates from the S strain, put it in with the R strain, and there's no transformation. Okay? So that was Griffith's experiment. I want you to go over it a little bit more in depth, but that's the general idea. Okay? So this is the link that I'm going to ask you to log on to. Okay? So you're going to go log on to this link here, and you're going to click on 1920, 1949, click on the transforming factor, Oswald and Avery. You're going to review Griffith's experiment, and then go through Oswald's experiments, and I need you to know this information from Oswald's experiment. Okay? So you can download the PowerPoint and then go through this, and these could be something that you'd add to your portfolio. Now the last thing we want to talk about is Hershey and Chase's experiment. So this was the concrete piece of evidence that definitively proved that it was DNA transferring our genetic information and not bacteria. Okay? Well, in order to understand Hershey and Chase, we need to understand these thing called bacteriophages. So you may have seen these in grade 11. This is simply a virus. It's a specific type of virus that we find very handy in biology because we use them. They're also called vectors or bacteriophages. And here's the neat thing. Remember that we're trying to figure out whether it's DNA or it's protein that is the genetic material. Well, when we take this virus right here, okay, the virus is composed of only two things, protein and DNA. Conveniently, the two things that could possibly be the hereditary material. So we've got DNA inside a protein coat, okay, and this whole thing is called a bacteriophage or a virus. And Hershey and Chase used this thing to identify, well, is it the DNA or is it the protein coat that's containing the genetic information? Okay. So this is kind of the big diagram, and I'm going to ask you to go through the experiment on that link that I gave you before, this link. Go through the Hershey and Chase experiment. Okay. We want to make sure that we understand it. I'll probably talk about this maybe a little bit tomorrow, but I want you to go through it at least once because if I go through it right now, it's going to be very confusing until you've at least looked it up once. Okay. So what I'm asking you to do is to go back to that timeline that you were here before, this website right here, and you're going to go through the Hershey and Chase blender experiment. I want you to kind of summarize the experiment. You need to be able to explain to me what exactly is happening in this diagram. Like, what are they radio-labeling? Why are they radio-labeling sulfur? Why are they radio-labeling phosphorus? Okay, and kind of explain to me what exactly is happening during the experiment. So those are the three things that I want you to go through. Okay, so you've got Griffith's experiment, which you need to know about, Hershey and Chase, and then Oswald Avery's experiment, and then Hershey and Chase. So I would get some diagrams out. If you go to Google Images, look up Hershey and Chase experiment, Oswald Avery, and the transformation principle, and if you can explain what's happening in that diagram to a certain level of detail, and that's the big point of today, is discovering how exactly we knew DNA is the genetic material. Okay? Email me with any questions. Okay? Tomorrow we're going to go on to structure of DNA, which goes back to content. I'll probably have you do a little bit from the content section, and then I'll upload the lecture as well. Okay? We have to do a little bit of lecture in this part because